students, this is Dr. Antara, your chemistry mentor, welcoming you for one more session in surface chemistry. Today in this session we shall see the characteristic features of enzyme catalysis. Okay. So before going into the topic, let us see what are enzymes first. Actually, enzymes are complex nitrogenous organic compounds produced by plants and animals, right? See, several chemical and biological reactions occurring in the animals and plants are catalyzed by some important group of globular proteins called enzymes. Okay, so these are the nitrogenous compounds. Enzymes are the nitrogenous compounds produced by plants and animals, and they are protein molecules of high molecular mass. Okay, they have high molecular mass and they act as effective catalysts. Okay, see, without these enzymes the living processes will be too slow to sustain the life. Okay, for example, in the absence of these enzymes in our digestive tract, it would take us about 50 years to digest a single meal. Thus, enzymes are absolutely essential for life processes. Understood, students? So these enzymes, they catalyzes numerous natural processes in living bodies, and that's why these enzymes are called as a biochemical catalyst. Okay. To conclude in a nutshell, see, these enzymes are the important class of globular proteins which acts as a biocatalyst, okay, in the living systems and are produced by the living cells, okay. And there are certain examples for these enzymes actually in stomach. Pepsin, see, this pepsin is an enzyme which converts proteins to peptides. Understood? And the pancreatic trypsin. So trypsin is another enzyme. This converts proteins to amino acids by hydrolysis. Understood? And there are so many other enzymes also. For example, urease, invertase, okay, etc., etc. These are the different types of enzymes which acts as catalysts in biological processes. Understood, students? So let us move into the topic now. The first characteristic features of the enzymes is that they are highly specific in nature. See, the enzymes are highly specific in nature. Each enzyme is specific for a given reaction. That is, one catalyst cannot catalyze more than one reactant. That means one enzyme cannot catalyze more than one reactant. Understood? That's the meaning. Okay. That means they are highly specific in nature, right? See, since the action of the enzyme is highly specific, every biological reaction requires a particular type of enzyme. Okay. See, to up to date, there are more than 3,000 different kinds of enzymes. So, how many enzymes are there? More than 3,000 types of enzymes are there, have been identified in specifically in living systems. Okay, each catalyzing a different biological reaction. Okay. So enzymes are highly specific in their actions on substrates. Why? Because of their shape. They have a specific shape and size. Because see, these enzymes will be having some cavity or the active sites on their surface, and only a particular substrate can fit into that active sites. Okay, that's why these enzymes are said to be highly specific in nature. For example, the enzyme urease, it catalyzes and hydrolysis of urea and not other amines. Okay, it catalyzes and hydrolyzes urea and not other amines. Here, see, this is urea and this is hydrolyzed by this urease. Okay, see, urease. This is catalyzed and hydrolyzed to form ammonia. But the same urease, here we are making use of the same urease to hydrolyze N-methylurea, but it is not possible. There will be no reaction taking place. Since because this urease is very specific and it can hydrolyze or catalyze only urea and not the other amide. Understood? The next important point is they are highly efficient. Mean one molecule of an enzyme may transfer 1 million molecules of reactant per minute. That means one molecule of an enzyme, it has the capacity to transfer 1 million molecules of the reactants to products within a minute. Okay, that's why these enzymes are said to be highly efficient. They speed up the reactions up to 10 million times actually. This enzyme can speed up a reaction up to 10 million times as compared to the uncatalyzed reactions. Okay, so when you are comparing this catalyzed reaction of enzymes with that of uncatalyzed reactions, this catalyzed reaction, that is the reaction which are catalyzed by these enzymes, will be undergoing 10 million times higher speed than that of those reactions which are uncatalyzed. Can you understand? Right? Okay. Let us move on to the next important point, that is they are highly active under optimum temperature. these enzymes are highly active under optimum temperature. What do you mean by optimum temperature? See, it's a definite temperature at which the enzyme reaction will be maximum. Okay, see the rate, what is optimum temperature? The rate of an enzyme reaction becomes maximum at a definite temperature called optimum temperature. Understood? On both the sides of the optimum temperature, the enzyme activity decreases. That means, see, below this optimum temperature, the enzyme catalyst reactions are slow. Understood? See, below this optimum temperature, the enzyme catalyst reactions are slow, and above this temperature, the enzymes get denatured and they will be losing their activity. Can you understand? So, this the optimum temperature is a particular temperature. For example, if it is in a human body, the optimum temperature will be 37 degrees centigrade. Okay, 37 degrees centigrade. That means 310 Kelvin. 310 Kelvin. Okay, so this is the optimum temperature. The optimum temperature range for enzyme activity is 298 to 310 Kelvin. So within this temperature, the enzyme activity will be very high. But below this 298, what happens to this enzyme's activity? It gets reduced. Okay. But above this temperature, what happens to the enzymes? They get denatured and they will be losing their activity. That's the meaning. Okay. So the human body temperature being 310, it is suitable for enzyme catalyzed reactions. Can you understand? So this is the optimum temperature for human body for the enzyme reaction to take this properly. Okay. Understood, students? The next important point is they are highly active under optimum pH. See, just like that of optimum temperature, the pH should also be a particular, pH. The medium should be having a particular pH. Okay. Actually, the effect of pH on the activity of the enzyme is little complex. However, the optimum pH for most of the enzyme is around 5 to 7. Okay, that's why they have fixed the pH, optimum pH for the enzyme catalyst reaction as 5 to 7. Understood? So between this pH, the activity of the enzyme will be maximum. That's why this pH is called as the optimum pH for enzyme catalyzed reaction. Understood, students? The next important point is increasing activity in the presence of activators and coenzymes. Okay, see, the enzyme activity will be enhanced in the presence of activators and coenzymes. Let's see what are coenzymes and what are activators. Okay, see, actually, the enzymatic activity is enhanced in the presence of certain substances known as coenzymes. Okay, see, this even a small amount of vitamins would enhance the activity of these enzymes. Okay, actually, these vitamins will be acting as the coenzymes. Understood? So what happens in the presence of these coenzymes? These enzymes activity will be enhanced. Okay. So for example, what are the examples for these coenzymes? Actually, adenosine triphosphate (ATP) you would have heard while learning about a DNA, RNA. Okay. So there you might have learned about this adenosine triphosphate. Actually, this acts as a coenzyme, and vitamin B also acts as a coenzyme. Okay. These two will be, and we are having many other coenzymes also. Those coenzymes will be enhancing the activity of the enzymes. Understood? Next comes the activators. Actually, activators are generally metal ions. Okay, see Na plus ion, sodium ion, manganese, cobalt, copper. Okay, so the ions of these metals will be acting as the activators for the enzyme. Actually, these metal ions are weakly bonded to the enzyme molecules and increase their activity. Okay, what is the mechanism of these metal ions or the activators? They will be weakly bonded to the enzyme molecules and thereby they will be increasing the activity of the enzymes. Okay, actually amylase is an enzyme. Actually, amylase in the presence of sodium chloride will be having an enhanced activity. This amylase will be having an increased activity due to the presence of sodium chloride. So the sodium metal present in the sodium chloride will be acting as the activator for this amylase enzyme. Understood, students? Next important point is influence of inhibitors and poisons. Okay. So previously we have seen about the activators and the coenzymes, and here we are going to see about the inhibitors that inhibits or which decreases the activity of the enzymes. Okay. So the inhibitors or the poisons interact with the active functional group on the enzymes surface. So the enzymes will be having the active functional groups on the surface, and what happens is this inhibitors on the poisons, they'll be interacting with the active functional groups on the enzyme surface, and either they will be they reduce or completely destroys the catalytic activity of the enzymes. Can you understand? So what is the role of these inhibitors or the poisons? They will be interacting with the active functional group of the enzymes, and either they will be reducing or they will be completely destroying the catalytic activity of the enzymes. Okay, actually the action of the enzymes are controlled by various mechanisms, and they are very sensitive to inhibitors. Okay, actually these enzymes are very sensitive to inhibitors. Okay, actually certain drugs we are making use of, right? When we are sick, we are making use of certain drugs. These drugs will be acting as inhibitors for the enzymes. Okay, so the usage of many drugs even can be acting as an inhibitor, and various organic and inorganic substances such as HCN, H2S, C2S will be acting as inhibitors for the enzyme. So if it is present in our body, they will inhibit the activity of the enzymes. Okay, so in the presence of these substances, enzymes lose their activity. Understood, students? Might have understood about the inhibitors and poisons. Now, okay, the last one important characteristic features is even a very small quantity of enzyme is sufficient for a particular reaction to take place. Okay, see here, a very small amount of enzyme can be highly efficient. See, they have given an example, enzyme renin. Okay, enzyme renin can coagulate over 1,000,000 times of its own weight of milk protein. Can you understand? So even a small quantity of enzyme renin is enough for coagulating 1,000,000 times of its own weight of milk protein. Understood? So this is because the enzymes molecules get regenerated during their catalytic activity. Can you understand what is the reason why little quantities required for for this particular reaction? The reason is this particular type of enzymes molecules, they get regenerated during the catalytic activity. So a typical enzyme molecule may be regenerated 1,000,000 times in 1 minute. So it is possible to regenerate itself 1,000,000 times in 1 minute. Okay, so that's why a very little quantity of enzyme is enough for a particular reaction to get catalyzed. Understood, students? Well, fine, students. I hope you might have understood what I have taught today. Let me meet you with another important topic in my next session. Until then, it's Dr. Anita Raj, your chemistry mentor, signing off from you. Thanks for watching.